Unfortunately wasn't able to make it to be here in person, so we have a recorded session from him. This is David Yeany, and the talk is titled Swainson's Thrush Populations and Habitat Use in Pennsylvania Old Growth Hemlock Forests, so a follow-up on Mary Ann's talk. David is an avian ecologist for the Pennsylvania National Heritage Program at the Western Pennsylvania Conservancy. He has 19 years of professional experience in ornithology and bird conservation, holds an MS in applied ecology and conservation biology from Frostburg State University, and a BS in biology from Messiah University. His work focuses on bird species of greatest conservation need, including surveys and monitoring, habitat analysis, and movement tracking. He serves on several state and regional bird conservation and technical committees, and he co-leads the International Evening Grosbeak Working Group, formed as part of the Road to Recovery initiative. And I'll just take a quick moment to get it up, and then we'll get that one going.
Good afternoon, everyone, and thank you for joining this session. Today, I will share our work in Pennsylvania on Swainson's thrush populations and habitat use in old-growth hemlock forests. This work was done in collaboration with my colleagues Lucas DeGroat from Powder Mill Avian Research Center and Mary Ann Peretti, a fellow scientist in the Pennsylvania Natural Heritage Program, who just presented on our wider old-growth forest project in Pennsylvania. First, I would like to acknowledge the organizations and funding sources which contributed to the work I'll present today. In particular, funding from a state wildlife grant, Pennsylvania Department of Conservation and Natural Resources, and the Pennsylvania Natural Heritage Program. Also, I offer a special thanks to Joel Throckmorton for his work on the field components of this project. Swainson's thrush is a neotropical migrant that breeds across the boreal forests of Canada and northern United States, montane and coastal forests of western North America, and the conifer forests of the Appalachian Mountains. Here in eastern North America, we have the olive-backed or Swainsoni group, which includes our Appalachian subspecies, which evidence suggests spends the non-breeding season from Colombia south to northeastern Peru. In Pennsylvania, Swainson's thrush is at the southern edge of its eastern breeding range. Swainson's thrush is one of Pennsylvania's rarest breeding songbirds, with exact estimates uncertain. However, Partners in Flight currently estimates the Pennsylvania breeding population at about 3,700 individuals. Swainson's thrush is experiencing range-wide population declines, as has been visualized in the eBird breeding trends map shown here. Particularly large declines appear in the northeast and northwest, but decline is also shown across the entire central boreal forests of Canada. Similarly, breeding bird survey data show a 0.43% decline across the continent since 1966. Though these surveys do not adequately sample the species and provide some unreliable trends regionally, so we need to look to other sources. The Vermont Center for Eco-Studies has been monitoring Swainson's thrush and other mountain birds and provided some current trends in their State of the Mountain Birds 2024 report. This covers data from Maine, New Hampshire, New York, and Vermont. Their graph shown here shows local population size declining across this region consistently with some inter-year variation. But they found an overall 2.24% decline for Swainson's thrush and a total decline of 27.2% from 2010 until 2024. In Pennsylvania, Swainson's thrush is a special concern species which is tracked by the Pennsylvania Natural Heritage Program and ranked as an S3B vulnerable species. And it is also a species of greatest conservation need, listed in the 2015 Pennsylvania Wildlife Action Plan. It's also worth noting that Swainson's thrush will also be included in the 2025 Wildlife Action Plan update. Swainson's thrush is most threatened during the breeding season by invasive forest pests, forest fragmentation and degradation, and climate change. Based on eBird breeding trends maps for Pennsylvania, we can see strong declines across much of the species' limited range in the state, especially where Swainson thrush is most abundant here in the Allegheny National Forest region. This results in an eBird estimated overall decline of 10% from 2012 to 2022. The focus of our Swainson thrush work in Pennsylvania has been in the western population located in the Allegheny National Forest. This map shows breeding populations in orange across the state with the Allegheny National Forest in gray. The Allegheny National Forest is where we have documented the highest densities of Swainson thrush in the state. In 2015, the Pennsylvania Natural Heritage Program conducted bird and vegetation surveys across the north-central region of Pennsylvania, known as the PA Wilds, shown in black here. This project focused on determining important habitat factors for a number of forest interior bird species. For Swainson's thrush, its highest density was found in hemlock forest when compared to northern hardwoods and oak forest types. And two habitat factors had a strong positive effect on density: tree canopy height and woody debris. Swainson thrush appeared to benefit the most when tree canopy was greater than 20 to 35 meters in height and woody debris cover was greater than 20% all the way up to 40%. In 2019, we implemented a tracking study on Swainson's thrush in the Allegheny National Forest, looking at microhabitat use. We deployed low-tech radio nanotags on 21 male Swainson's thrush in June and July of that year. We did on-the-ground tracking through August and relied on MODIS stations for post-breeding and migration tracking, but I won't be discussing that component here today. We also collected forest vegetation measurements at 4 points within each mapped Swainson thrush territory. Our main objectives were to, first of all, map the breeding territories of these birds, then to determine the use of hemlock forest and other habitats during breeding, post-breeding, and migration, and, as I mentioned, track migration routes southward from Pennsylvania. We completed this work at 3 sites within the Allegheny National Forest. All 3 of these are shown by red stars on this map. The first is Heart's Content, a recreation area with a small patch of old-growth hemlock white pine forest. The next site was East Branch Tynesta Creek, a small riparian patch of older hemlock forest with higher levels of human disturbance, including dirt road and campsites. The last site was the Tynesta Natural Area, which exists as a 4,000 plus acre patch of old-growth hemlock white pine forest, and is the largest old-growth patch between the Adirondacks and Great Smoky Mountains in the east. You also see on this map existing MODIS receiver stations in 2019 in yellow with their antenna ranges also shown, overlapping some of the sites. We used low-tech nanotags with 2 different burst intervals of coded clicks at 19.1 seconds and 11.3 seconds. These different burst intervals, with one nearly double the other, did present challenges at times when tracking on the ground. However, by using tandem receiver tracking with 2 receivers, we were able to more quickly triangulate a bird's position and acquire more accurate locations. While shorter burst intervals would have made ground tracking easier, we wanted to ensure tags had enough energy to give us migration tracks as well. The 19.1 second tags even provided us enough tag life to add the potential of spring migration detections the following year. Now, unlike automated radio telemetry tracking using new networks of nodes and receiver stations, we collected data manually based on handheld receiver detections. We collected positions using handheld Garmin GPS at a minimum of 5 minutes apart. Locations were captured as close to the known or estimated position of the bird as possible, and distance to the bird was estimated to the nearest 1 to 5 meters as much as possible. We also recorded behavioral notes, visual or auditory confirmation, as well as signal strength and gain. In total, for 21 Swainson thrushes, we recorded 386 detection locations. For 126 of those locations, we had field-based detection distances. We examined the relationship between detection distance and signal strength and found a significant linear relationship. We then applied the equation for the 80th quantile of that relationship to estimate detection distances to 260 locations without field-based distances. This resulted in a mean estimated field distance of 26.8 meters with a range between 10.2 and 39.1 meters. And this allowed us to produce detailed territory maps for hemlock forest used by Swainson's thrush. Over the next few slides, I will show the Swainson thrush territories we mapped using those estimated detection distance buffers. First, we have 4 birds from the Heart's Content site. The bird shown on the left here, Swainson's thrush number 184, had the largest territory measured at this site. And we also captured a fall migration track for this bird via MODIS. This next map shows territories for 11 birds tagged in the Tynesta Natural Area. This area holds the highest density of Swainson's thrush in Pennsylvania, and as you can see, we found that 8 out of the 11 birds actually had overlapping breeding territories. We hypothesized that these birds were likely cramming into the highest quality habitat, thus the overlap of territories. Finally, we had 5 territories from Swainson's thrush at the East Branch Tynesta Creek site, where we had older hemlock forests with more anthropogenic influences. On the left here, we have Swainson's thrush number 187. This was a bird that held a consistent territory at the north end of the site, where we were able to locate its nest in 2019. We also detected a northbound spring migration route for 187 in 2020 and followed up with surveys to find him on his same 2019 territory. Now we arrive at the crux of our microhabitat tracking study of Swainson's thrush. Within each of those territories that I just showed, we measured microhabitat at 4 randomly selected locations with detection accuracy of 25 meters or less. Now let's take a look at microhabitat data that we collected from 20 of those 21 Swainson thrush territories. We compared 11 forest metrics between 80 plots across the Swainson thrush territories with 53 unoccupied reference plots within non-old growth hemlock forests from across the Pennsylvania wilds collected during our 2015 project. We found 10 of these metrics to be statistically significant differences, and those are all shown in bold. Shown in red, Swainson thrush territories, when compared to reference hemlock forests, had greater bryophyte cover, woody debris cover was also higher, and tall shrub cover was also higher, as well as taller overstory height, which was 20 to 35 meters versus 15 to 20 meters at the reference sites, as well as more standing snags. Conversely, Swainson thrush territories, when compared to the reference forest, had lower basal area, lower overstory cover, lower subcanopy cover, and lower short shrub cover, as well as shorter subcanopy height in the 5 to 10 meter range versus 10 to 15 meters. Along with these metrics, we looked at characteristics of old growth forests related to tree size. These territories, occupied by Swainson's thrush, contain very large trees, particularly hemlocks, and we aim to compare these against other reference data sets. Unfortunately, we didn't have those same tree data within the reference hemlock forest. With the current old growth characterization project the Natural Heritage Program is doing in PA, which Marianne just presented on, we had the opportunity to survey breeding birds at more older forest sites. So point count surveys were done at 96 point locations across 4 sites in 2024. This included Heart's Content and Tynesta Natural Area in the Allegheny National Forest as well. While we are still analyzing these data, we do have some early results. We detected 74 bird species across the 4 sites, and 95% of survey point locations were within hemlock hardwood forest. In the figure on the right is a glance at the top 30 most abundant bird species within these sites, which also comprise 95% of total bird abundance. Among these species were 22 forest interior species. We also had 8 species that were old growth associates, including Swainson's thrush, and finally 3 woodpecker species, which were among the most abundant birds. Woodpeckers, of course, are known to have higher densities in older forests, having more tree snags. Probably most importantly, this current project allowed us to compare Swainson's thrush population data from 2015 to 2024. We compared data across 35 point locations surveyed in both years with the same point count methods during June and July. Looking at maximum detections per point, we found a total of 15 Swainson's thrush detections in 2024 across 10 point locations, versus 40 detections across 26 point locations in 2015. Via Kruskal-Wallis non-parametric test, we found this difference was significant, and it amounted to about a 62% decline over 9 years for the Tionesta natural area population of Swainson's thrush, which we sampled. In closing, I want to highlight some conservation and management implications based on our work, much of which aligns with those presented previously by Marianne. It's apparent that old-growth hemlock forests are essential to Pennsylvania's breeding population of Swainson's thrush, and they need to be protected to help these birds persist. We've shown the importance of maintaining old-growth forest conditions. In particular, Swainson's thrush need well-layered vegetation strata, tall trees, higher levels of woody debris, and proper consideration to both canopy and tall shrub cover. They'd likely also benefit from increasing hemlock recruitment in the understory. Lastly, we need to focus on the health of our old-growth forests and continue to address threats from hemlock woolly adelgid, beech diseases, and human-caused disturbances and other introduced invasive plants. Not to mention, additional planning should be made for climate change impacts and considerations of climate-related stressors and the potential for the need to replace hemlock with conifers that are more tolerant to a changing climate. Wrapping up, I'll just point to a few next steps for our old-growth forest work as it relates to Swainson's thrush and the old-growth bird community in Pennsylvania. We will take a look at changes in vegetation data from 2015 to 2024 to see if any correlations exist that may be tied to the decline of the Tionesta natural area Swainson thrush population. However, as noted earlier, with significant regional declines also happening, there are likely other factors at play that may occur during other points in the bird's annual life cycle. Nevertheless, continued monitoring in Pennsylvania will be needed for this species. This also would allow us to highlight key microhabitat factors that could be managed for Swainson's thrush and which could be easily monitored. Lastly, we will finish analyzing our new bird data from sites included in the characterization project for old-growth forests across Pennsylvania. And with that, I hope this presentation was informative, and I look forward to hearing from anyone who has questions or comments. You can reach me at my email address shown here, and you can also check out more about the Pennsylvania Natural Heritage Program's work and the Western Pennsylvania Conservancy online at these web addresses below. Thank you.